hardware, open access journals, open data for science. And I really do think that open hardware is an essential next step that will really help science in the long run. I'm not going to start talking about biology. Take a detour, and we're going to go to Switzerland. This is CERN. They do nuclear research here. And the reason I'm talking about that is one of their projects, most recent one, I'm sure some of you may have caught something, at least in popular media, about the Opera project, which was a project started in 2006 to study the oscillations of neutrinos and try and discover some transition between different states of neutrinos. Anyway, in 2006, they started construction. In 2009, they started collecting data just to calibrate the instrument. In 2011, they published a paper basically saying that they couldn't calibrate the instrument because, despite their best efforts, neutrinos showed up too early. They showed up 60 nanoseconds too early, and they spent 2 years going through every single piece of hardware in that chain from Switzerland to Italy, because that's where the detectors are at, to try and find and actually pinpoint how much error there was in every single step. And I love this one paragraph. It's near the end of the paper, basically saying that despite all our work, we can't figure out where this error comes from. And we're throwing it out there to the science community to see what they think. Because obviously this is, if it holds up, it's an astonishing experiment, an astonishing result that truly challenges the very pillars of modern physics. Because the speed of light should be unbreakable. Right? And this is, I wanted to talk about this because, extraordinary claims like that, like challenging 80-year-old scientific paradigms, can only be done with extraordinary evidence. Right? And that's a paraphrase of Richard Feynman's quote. And open hardware was a big part of that effort. CERN and actually the entire physics community really uses open hardware pretty much exclusively, but they've developed a good collaboration with companies. And this is a slide from a presentation on their website. The benefits to the open hardware is they get to design exactly the way they want the peer review. So now they get a chance to have peer review of the designs, as well as the peer review by scientists as well as corporations. The reuse is dramatically improved. And the last one is a pretty important one, actually. There's no vendor lock situations. And you're not tied into vendors. These are a lot of the same benefits there are to open source software, and it should be understandable. And it's just nice to see people getting these benefits from the hardware as well. Now, the hardware that they're developing it's as integrated as is being developed by corporations as well. And so this is that they have a single board Linux computer. It's under $50. Cost. So you can reasonably have this assembled for $100. Other fields where people started using open hardware. This is a scientific CCD sensor. It's modular, so you can drop in different actual sensors into it. This is used, in the Global Hawk unmanned drone that's operated by NASA uses a CCD camera that this modular hardware. also the same camera, the same basic technology that's the heart of building the Google Street View image database. Um, and I just want to make sure you guys understand that open hardware is not something new. It's established and it's used by, well, I mean, even NASA and CERN. So it's perfectly reliable. Biology, on the other hand, chooses to go a different route with this. We largely rely on commercial tool companies. Biology's developed a working relationship with industry. We start the creation of ideas and then we hand them off and rely on a commercial company to commercialize the product. And science makes a big investment in tools. I mean, biology specifically. We invest dollars, money, to purchase these machines, which can cost upwards of $250,000. But more than just the money is the scientific capital, right? Because no machine's perfect. And a lot of research goes into developing the error profiles and incremental improvements, which is what science is all about, right? Improving on the last researcher's work. Science has really begun to rely on, and this one specific vendor specifically, Lumina, on sequencing. I read in a journal that 90% of sequences done today in academia are produced from Illumina hardware. And this has really been driven by the emphasis on genome-wide association studies, GWAS studies. Tons of publications showing how genetic polymorphisms are really directly tied into a lot of clinical phenotypes. And this is part of the reason why there's so much effort by scientists to improve the methodologies employed in these instruments. Right? And so this is an example of some of the stuff we're doing. This is pretty exciting, right? I mean, starting to find the genetic signatures, basically the genes involved in longevity, which is great until this happens, right? The dreaded unpublished data. So this had to have been a terrible feeling. The unpublished gotcha, which apparently was known by some investigators, but not by the particular authors of this paper that used this Illumina array. Eventually, the paper had to be retracted because it turned out it was the entire result of the paper was artifactual in nature. Right? No one's immune to these bugs, right? The same bugs played bioinformatics, hardware developers as well. And the investment science makes in keeping these instruments reliable is, that's why we do it, which is great until some recent events. I don't know, just a couple weeks ago, some of you that may have been following the news may have noticed that Roche is engaged in a hostile takeover of Illumina right now. And what this means, there's a lot of, we don't know. There's a great Nature editorial on it and what it could entail for the company. Here's some quotes to kind of put this into perspective. Ultimately, Roche may or may not change anything that's going on in Illumina, but certainly there's going to be layoffs, teams will be disbanded, and we don't know what will happen to this platform that science has invested so much money and scientific capital on to use as basic infrastructure for the genomics revolution. Personalized medicine, all of this relies really on these basic research tools being available to scientists, not just the doctors and big pharma. And so I want to talk about where we at today with open hardware and biology. And so I'm going to start actually with the role of scientists develop tools and a really big emerging trend, a really big trend in biological hardware, which is microfluidics. So microfluidics offers the ability to automate scientific experiments like wet bench experiments, which improves the reproducibility and can facilitate some high throughput approaches. It also, the micro part of the name is the smaller scale. They take advantage of the laminar flow. The properties of liquids are very different at a microscopic scale than they are at a macroscopic scale, and that gives a lot of leverage to improve the sensitivity and the sensitivity of assays. There's also less use of less reagents, and this is important when dealing with primary cells like clinical samples. We can only isolate a small number of cells, and scientists, tool developers, right? And a lot of them have used microfluidic techniques to do some pretty amazing stuff. I mean, they've done single cell ELISAs, single cell QRT PCR is so for the non-bio. The single cell ELISAs, the top one is being able to detect at the single cell level what proteins that cell is expressing. Single cell PCR is being able to detect at the single cell level what genes a cell is expressing. And then flow cytometry is automated sorting, so identification and sorting of cells, right? And there's entire journals devoted to these types of publications, which are largely proof of concept papers, demonstrating the capability. And this is the general pipeline, right? You generate the proof of concept, maybe develop the engineering a little bit more, and then hand it off to a for-profit company for further development. So fluidics. So here I show this is a schematic. The dark blue areas of the drawing show flow is taking place. The light blue areas are areas of stagnant or stagnant where there is no flow. The red boxes and the green boxes are the state of valves. So some of the one approach to automating microfluidic experiments is to incorporate valves, and that you use to steer flows. So you can see by opening up different valves, we can flow direction and control where we, in this case, are applying the cells and where we're directing and steering reagents. Here is a movie showing the little crossed area here is what we're looking at. And what you see moving in there now, the cells. So we're loading the device with cells, and we open up some valves, and now we're going to clear this channel of all the cells. And these pads here are valves. And so now we can close these valves, isolating each one of these chambers, allowing us to treat these cells in different manner. And that's the basic principles of microfluidics. There is no universal chip that does they're called chips, by the way, the devices. The microfluidic devices are traditionally called chips just because of the close ties between how microfluidic devices are manufactured and how semiconductors are manufactured. So the microfluidic devices, there's no killer design that can do everything. Every chip design is basically the physical manifestation of a specific protocol. Right? You need a different chip design for every assay. And given the wide range of assays, it's reasonable to expect we're never going to have very high volume need for very high volume any one design in science, basic research. So in one of these designs that was in the paper published by our lab 2 years ago, we had to design a chip that had 6 chambers in parallel, where we could apply 6 different solutions to each one of these chambers. And these are the chambers right here. And so we have to have 6 inputs for the 6 solutions, and the valves have to be ranged appropriately in the right number of inputs. But this is just one test, and a science paper needs, any scientific publication needs more than one experiment. So the second experiment was done to study the kinetics of calcium recovery and depletion. This required a chip design, one where we only apply a single solution chambers, but now we can do so in a time-dependent manner. Apply the solutions for different lengths of time in each one of the chambers, as opposed to the earlier design, which only allowed us to apply the solutions with identical timing. There are minor variations, but the reality of it is that it's very difficult to make these devices. And Robert ended up making, I believe it was 16 different versions of the designs for arriving at these last 2 that made it into the paper. And every design has different numbers of requirements. You see down here that this 17 channels at a higher pressure and 7 channels at a lower pressure, and the previous one is a 12 and 12 at the 2 different pressures. And this is pretty typical. And this starts driving some of the, were all things that I was thinking about when I got the project years ago, right after Robert left. So I was basically handed the equipment and asked to, get it working again. This was after the lab had moved, so everything had been boxed up. And it was really difficult. I'll give you an illustration of why. I mean, this is not our setup. This is a paper. This was published in a supplemental information of another microfluidics publication, but it's very exemplary of, what a microfluidics lab ends up looking like. It's really a lifestyle choice. It dominates the room. And you see up here, these are valve manifolds, solenoid valves. We have the computer to run it, custom-built incubators, pressure regulators. It's very involved. This is the paragraph describing the automation. This is in the supplemental information from this publication. And I highlighted the 2 phrases that coming at this for as an outsider, trying to reassemble it was what everyone does. And this is totally widespread. Custom electronic units and custom software with no more information other than that picture. Right? This basically makes independent reproduction without help from the author impossible. And this is not limited to a single lab. It's really widespread throughout the field. But there's a growing appreciation of the need to document and make it more reproducible. This is a different approach to doing this. By the way, this hardware is a slightly different approach than the pressure-driven are pneumatic pressure driven. This is hydrostatic driven, and so there's these linear actuators that move these syringes up and down to apply different amounts of force, hydrostatic force to the chips. Again, you get the general idea. Everything's investigator built. This is some of the best practices for doing documenting this. At least now, people are starting to, there's publications, chapters, and wikis linking to all this. But really, the wiki format and really the existing tools we have for sharing information like this aren't suited to sharing the type of data you need to really capture the essence of these kinds of hardware systems. And I'll be getting back to that in just a second. I'm going to take a little detour and tell you why scientists need to build this kind of complexity. So this is just sorry for the poor quality, but you'll see why in a second. This green box here represents the cell membrane. And along the cell membrane, we have these receptors on the surface that detect compounds in the surrounding media. In this particular, we're looking at a map of this particular receptor family that responds to bacterial infection and other types of immune markers. So not just, and up here we have, microbacteria, gram-positive bacteria produce these different compounds that bind or respond to different of these receptors. Boxes down here represent, this is an information network. So it's a node in the network, each box, right? This is the network that controls entry to the immune response, right? It's a overwhelming problem. They're trying to study this at this gestalt level is completely unmanageable. I mean, so consequently, scientists focus on small little segments, right? That reductionist approach, breaking it down into little modules and studying that. And keep in mind that this map represents the signaling, the information processing pathway of a single family of receptors. Right? There's hundreds of these types of maps and information networks in the cell, and each one of these networks is also linked to other networks, right? And so you can imagine a map of maps, right? It's these emergent properties that we want to study are difficult, and we need to have the kind of the tools that we have need to match what we really want to do, which is understand the biology, the cure disease. And so early on, I did a power analysis. And for those of you that aren't familiar with what a power analysis is, it's a way of quantifying the likelihood of success of your experiment, just from a purely statistical point of view, right? So if you're looking at the difference, if you're looking at testing means, like the average response, right? How much a cell responded, and the standard deviation, detect smaller and smaller differences means you start needing to detect exponentially more number of cells. If you want to detect proportions, like what fractions of cells responded to a treatment, you need to be able to image a large number of cells as well. And this is for a single condition, right? You can have multifactorial designs, say like you have 10 compounds that you want to study, but you need to study them at 3 different concentrations, and you're looking for subtle regulators. That would require imaging 90 1,000 cells, and at the current limitations of microscopy, that means you'd be looking at 107 runs over 214 days, because that takes about 2 days per run, right? And science right now tackles these projects by, it's difficult, and it's certainly not. The current way we do it is we just throw a postdoc or a grad student and try and get them to finish this out. But the reality of it is that this doesn't scale. Pharmaceutical industry needs to screen thousands of compounds. So the approach that they do is very different. They build out, pharmaceutical industry, these high throughput, fully automated robotic labs where everything is within reach of a robotic arm, and the robotic arms move samples around the lab. And this is all under control of multiple computer systems. The labs cost, $20 million, $30 million to build. And just like a microfluidic device, they're really good for one type of experiment because, everything involves moving around the plates in a certain order and pipetting a certain amount. Changing it requires, at best, reprogramming the lab, which is no mean feat. And at worst, totally reconfiguring the position of all the hardware because you need to bring in a new piece of instrumentation or something. This actually video came from a paper on the automation of science. It's actually a pretty cool paper. They in this paper they've linked the expert systems like artificial intelligence to run this lab, and they're using it to, the computer algorithms are deciding themselves what experiments to conduct, so which samples to carry on to the next stage of experiments and everything. So this is the supporting material from that journal article. It really is blank, literally, and I think that really says it all as far as reproducibility. So what I wanted to do is start over and make a system where, admittedly, I'll just tell you, laziness is the mother of all invention. I knew I couldn't be the person that sat in front of the microscope for 105 days straight. Not at this point. So I decided to spend my time re-engineering the system so that it was actually usable by, the people that work in science. The technicians and the undergrads, the students, right? Let me show you that system actually. I just need to switch computers for a second. All right. So this is a microfluidic, a schematic of a microfluidic device. It's an object-oriented design because I wanted to be sure to get some of the benefits of object-oriented features, the object-oriented methodology, inheritance, composition, aggregation, because it really does enable the reuse. So this circuit, we have at the bottom, we have the paths where the fluid flows. We have cell chambers, we have reservoirs, we have valves, and this is all described in these parts, right? Where this part is here. And so these parts are separate files, actually. This whole thing is an aggregation. This is device stack is an aggregation of different objects, like this fluidics object, which is an aggregation of separate, different distinct objects. And so by grouping objects in this way, it speeds up and simplifies the design of, this is the positive, so where I want all the circuits to be. So I get to design what I want. And then I can automate how to build it and how to interface it, which are two huge challenges in microfluidics, right? So this is the circuit. And now this is a template. This is a mold template. So I have these blank parts, right? Include all. You'll get to see exactly what this does in a second. But I combine these features with the templates to produce derived parts. Right? That combine the templates with these other objects, right? So it's still, we can work with standard objects, but simply by combining these objects in different ways, we can define the variety of design we needed to capture all the protocols that we'll need. So we still have that flexibility where users could define their experiment, but we're relying on standard objects rather than the way it's done now, which is everything is done as a 2D drawing in AutoCAD and then extruded. So there is really not an opportunity to reuse the geometry in any useful, meaningful way. Once I have these molds, the top and bottom molds, I can, this purely for visualization. This is what the chip looks like. I can also make the interface for it. This is the part that attaches and connects all these fluid lines to the world. So this is a chip to world interface, and this is of course integrated into the bigger I find it. And one last one. Right, so for visualization, we can now actually see the chip on the microscope. That we can determine fit and everything else. This gets us to one of the big challenges in this, in the design of hardware. In software, there's that critical problem. So how we keep track of the different versions. Just hide some of this stuff and get to. Okay. This will be kind of
so
informative.
Carla, I just wanted to say at this point that this is just so incredibly cool that you can essentially design out of standard objects that are physical entities and not what we typically think of as these software entities, that you just get to design physical hardware, and it's all stored as a file that you can then give to your curators or add as a supplementary table or what have you. And if anybody wants to have a microphone at this point, just to see this, just go ahead and raise your hand. It's the little raise your hand on next to your speaker and your microphone, and I should give you speaker rights. So we have another about 20 minutes. So I could start to basically just open this up to where questions are possible. Okay, let's go through the. Go ahead. I'm sorry. And I think you're getting applause from you're getting applause from the crowd.
airtight. It means airtight. You can do hypoxia research in it, and if be modified, these templates provide a starting point for another researcher, so they don't have to reinvent the wheel themselves. This is a Micromanager, which is a standard software for running microscopes. It's open source, by the way. Runs on Linux Macs, and so on the bottom you see the control screen. This is on the valves. This is the schematic, and so you can see that as we open and close valves, the flow changes. And so this is just a general purpose design, which means it's probably not all that useful to anybody since it's general purpose. But it's a start, and it'll let us collect the data that we need to show that this approach is going to be fruitful for other investigators. So some of these benefits, there's the whole DIY buy, which I actually did not realize existed as a movement until just a few weeks ago. But I'm really impressed by the quality of tools that are being developed by citizen scientists, not necessarily the establishment, right? So there's an open PR machine. You can buy a kit for $5. It's commercially built machines that cost $2,000 for like the no-name manufactured ones. Turn your iPhone into a decent enough microscope. This is useful. I mean, the applications that claim are developing world or just simple applications. You don't need a very high resolution microscope. There's this 3D printable labware. Even centrifuge for microtubes that runs off a Dremel tool. These are all, and this has already been done. It's just a matter, I think, of collecting all of this information and putting it in one spot, and getting the policymakers to fund this stuff. I mean, PubMed Central is valuable. I think engineering information is just as valuable as articles and data. Another big benefit to this is that resource divide in science. I mean, being at these doctoral institutions, we maybe don't realize that we're in the 1%. I think it's, what is it? The top 100 institutions in the country, like degree granting institutions, get 75% of all the NIH grant money, right? Which leaves like the vast majority of students with no access to cutting edge, not even cutting edge, modern scientific equipment. We're teaching our high school and most of our undergrads too, outdated science techniques using outdated equipment. Improving availability of open hardware designs is going to help out make a big difference in science education. Literally, these machines can be built by the local campus machine shop. This is Joe, who is worked at the Scripps Institute of Marine Sciences Development Center, the machine shop. These machines, open hardware things, need to be built by people that pay attention to what they're doing. You can do it yourself. It takes about an hour and a half to build one of these manifolds, or you can pay your local campus machine shop to do it for you, based off of like solid designs. And finally, the last thing that I'm going to talk about is maybe a little speculative, but the thing that really gets me excited about this. Right? So way back, people thought the way to build faster computers is to build them bigger. Now that the key to high performance computing is clustering a bunch of relatively low powered computers Beowulf clusters, and of course, if you have more resources, you build your server farms out of rack mountable like 1 units or blades. But the general principle of going parallel with a cheap computer rather than building a powerful processor applied into science as well, into biology, right? And so to develop an open platform for an automated microscope, right, actually allows me talking about building 1,000 of them, and so to do 1,000 assays, I just run 1,000 machines. That requires robust, high reliability connections. That infrastructure, 1,000 of these machines, the ability to transmit the hardware electronically really begins to open up the possibility of allowing allowing scientists to send experiments over the internet to be run remotely, in much the same way we send jobs to be processed in the cloud. And we're wrapping up. So, in some pictures, latest and greatest generations of designs. There's not a single machine on there costs less than actually maybe Ion Torrent's only $200,000. We got to be pragmatists at some point. Federal budgets are shrinking. That means research budgets are going to shrink, and fewer and fewer people are going to have access to the machines that we need to continue to propel science. Right? Corporations are vulnerable to outside forces, and honestly, it's just responsible to plan for the demise of a hardware vendor. Right? We can't lose all of that information to the vagaries of corporations. Right? And right now, instruments don't support this kind of collaborative multi-site efforts. And I think that this could be a real opportunity to start building instruments a new way. Physics, it really is informative. They work in close collaboration with corporations because they don't do it. But their sciences continues to be involved in the process of developing open hardware, and everyone.
Okay, so one person has raised their hands. A couple of people are clapping. I'm going to try to hope that you can all and open up your microphones. You should be able to now speak. So Derek Jacoby, your microphone rights should be enabled. Can you try to talk?
Yeah, can you hear me?
Yeah.
Hi, Carlo. Fantastic talk. I really enjoyed that. So I have been involved with BioCurious in the San Francisco Bay Area, and am one of the founders of a biohacker up in Victoria, BC right now, and we've done a lot of messing with 3D printing, but one of the questions I have for you is what the size and resolution of 3D printing capabilities you require in order to cast these microfluidics devices.
So, thank you.
Thank you.
So are there other questions? Okay, so I'm not seeing any other questions. I do have one though, just from the NIF perspective. We obviously want to celebrate, first of all, this kind of open innovation because I think it's absolutely fabulous. And we want to support it in any way that we can. And one of the best ways we find that we can support some of these really cool projects is just to let people know about them. I'd like to thank all of the participants in today's webinar. There were over 22 people that actually listened in, and I think that that's very good. Now, when we make this the data and everything else available, I mean, we can link to you in some ways, but do you anticipate that basically there will be a web space that you will be able to provide to people, or would you need help with that in order to kind of say, hey, I use this specific model, this specific design, here are all of the different parts for this paper, and then that could resolve to all of the specifications for the microfluidics devices. So that way, when you publish your paper, if it's in microfluidics, that you would just be able to, almost with a single click, go in and grab all of the specs and try to reproduce that experiment.
Right. Yeah. My dream is to be able to download everything you need. Right. Like from the tools, the little jigs that we make in lab, all that digitally. And it would be great to. I mean, no one lab can do it all, and like clearly, I mean, I know I need help from the informatics community in setting up that web space for it. I mean, I like the idea of like the digital object identifiers or some universally unique ID to assign to each object as it's published. And that way, we can resolve a lot of the confusion about like what parts we're using, and it'll really enable the reuse of parts more effectively because if as it gets pushed to publication, that's like the release form, and that's the object that we can build off of in other publications. So the publication that wants to use the incubator can reference it by some unique identifier.
Exactly.
And then
actually resolves to an object that you're not able to just buy if the company hasn't gone out of business, but you can actually create that object at any time. And that's just
right.
the coolest part of this whole project. So we'll try to help get these digital object identifiers and everything else ready for you as soon as you hit go and say, okay, this is ready for prime time.
Yeah, we're working on the where to publish best. I mean, I'm all for publishing like in PLOS One because I love the idea of open access.
Mm-hmm.
And I'd much rather just get this out there quicker rather than waiting the usual publication delays. I mean, it would benefit me personally too, because some personal time restraints. But yeah, so I'm looking to get this out ASAP within a month or two, hopefully.
Excellent.
And PLOS One's really fast, so.
Yes,
Yeah, there's so much that needs to be done. Like, yeah.
Trouble getting her microphone hooked up, and I've got her on the phone. So what was the question? I'm sorry. Right.
Yeah. Is there a question?
Uh, yep. Yep.
Oh, let me just say, in the interest of full disclosure, the everything that we talked about here is, of course, property of the University of Regents, and they've elected to file patents, which is another uh, challenge slash opportunity, I guess. And that's legislative requirements. They're required to file patents on intellectual property they generate from federally funded money, um, which is an interesting problem. And we can also take lessons on how the physics community has dealt with that through licensing. CERN has an open hardware license to be working for them, and that's an area that we need to work at too. Anita, you still there?
Your sound cut out for a minute, but I'm back anyway.
Okay. Emka? Oh. I guess we lost. Is
I'm glad we got through the talk on it before it got lost.
Can you?
yes.
Sorry, Jay, I'm on. I have a question. Is there a central repository for this slideshow? And secondly, I'd like to know your thoughts on how this kind of open source hardware could affect research in the developing world, where access to equipment is even more challenging than it is for research facilities here.
So there will be a permanent link to this webinar. Anita's recording that. I'm not sure what that link is, but you can probably check back to the same site, the same announcement site. You can email me as well. Now, the question about developing hardware in developing countries. So, something that I'm very cognizant of, and that this is not as accessible as some of the other approaches, right? The lower cost, more accessible approaches that are being developed and pursued by other scientists. Like, I should finish some of the slides with bio. But ultimately, 3D printers are going to continue to drop in price, and this is where the benefits of an open hardware really start taking off. Because there is a flourishing movement for developing. There's actually grand challenge out now for making a 3D printer that can reproduce itself. That's actually, I don't know how far away that is, but that's really we can have a machine reproduce itself, and that machine could also be used to make hardware like this. This I can easily see this starting being getting adoption at universities,
Mm-hmm.
even in developing countries. Cost of 3D printers is coming down. Right now, the 3D printer we use is about $30,000, and that's and I know that I could probably get away with buying a less expensive printer, but I just want this one to have a machine shop here. Ultimately, having open access to this kind of stuff is going to make it more accessible, because you can pick and choose the features that you do, and maybe not everyone will be able to do every experiment, but at least um, that potential is there.
Oh, thank you.
Does that answer your question?
Yes, it does. I was involved in putting together an event called Maker Fair Africa in Cairo, and we actually had people from a DIY biology lab in New York called GenSpace um, speak to students at the American University of Cairo about the opportunities within the area of open hardware. So it's something that we find very interesting and potentially um, extremely enabling. And that's why I was asking the question, because I think
Yeah, and
many of us think that it has a huge amount of potential.
what's really going to make that, I think, transform that potential into like reality is um, uh, once we can start using the same hardware. DIY Bio is, I think it's great, but the reality is I don't think it'll get accepted by mainstream science. Like, in other words, like uh, most scientists, look, the neutrino detection experiment, when they detected that things flew faster than the speed of light, if that hardware had looked like a DIY Bio hardware does, I mean, not all of it, but a large extent of it, or like the way our current microfluidics hardware looks, no one would believe it. It would be dismissed as an artifact of hardware, right?
Mm-hmm.
And that's the real benefit, is when you start having this peer-reviewed designs that even the most cynical scientists are using as well. That deflects all of that criticism, and that's how you can get scientists, even from here in the United States, the PUIs in the community colleges, or scientists in universities abroad that don't have access to the $200,000 machines. That's how you're going to get them involved.
Well, thank you.
My pleasure. Thank you for listening. I know it was a long talk.
Okay, thank you very much, Carlo. I'm sorry I had a little brief lapse there. My computer crashed, but thank goodness you were able to keep going.
Yeah.
So again, thank you all for listening, and thank you so much, Carlo, for presenting this wonderful system to us. And
And
we'll let
are you
you.
going to post a link on the website?
Yep, I will
The link to
absolutely post it on the website. And I will send you a link to it. So we'll probably also tweet about it and all of those other things. And when you get ready to publish this paper, if you need a DOI or anything else, let us know. We will help you do all of that. We'll grab this repository and others and try to describe them in a uniform way so that when other people are looking for it, they will find it.
Yeah, discoverability. I mean, I've looked for hours for like for information about like open hardware in biology, and even at the end of that hour-long searching, I was still finding occasionally a site here and there. So yeah, we need better discoverability of this information.
Absolutely, and that's actually one of the reasons why we put all of our resource catalog entities onto the wiki, because Google loves it. So, but anyway, all right. So again, thank you very much, and thank you all for. Unless there's another question, are there any other questions? Okay, no. And for everyone on
Somebody
the.
has a raised hand, by the way.
What was that?
Somebody has a raised hand.
Yeah, we seem to have a raised hand. Somebody may have raised it. This is Amika. Does Amika?
Ask the question.
Okay.
I asked the question already, so my hand is no longer raised. I'll lower the
Okay,
hand.
perfect.
Sorry.
All right, fabulous. Well, thank you all very much again for attending, and have a good rest of your day.
Thank you.
Okay, thank you.